In this video, we will cover the differences between prokaryotes and eukaryotes, as well as the basics of endocytosis and the endosymbiotic theory. Biologists have grouped cells into two main types, prokaryotes and eukaryotes. Prokaryotes were the only form of life on Earth for millions of years before eukaryotic cells evolved. The main difference is that eukaryotic cells have membrane-bound organelles, such as the nucleus, while prokaryotic cells do not. There are also many differences in terms of cellular structure that we will now go over. As aforementioned, eukaryotic cells have a nucleus, while prokaryotic cells do not. Eukaryotic cells have multiple chromosomes, while prokaryotic cells do not. Eukaryotic cells are generally multicellular, while prokaryotic cells are usually unicellular, though some cyanobacteria may be multicellular. A big difference is that eukaryotes have a membrane around the nucleus, while prokaryotes do not, and instead have their DNA in the cytoplasm. Eukaryotic cells also have endoplasmic reticulum, Golgi apparatus, mitochondria, and chloroplasts. Eukaryotic cells can be found in animals and plants, while prokaryotic cells are found in bacteria and archaea. Endosymbiosis is a theory that eukaryotes evolved from prokaryotes. How though? How could a eukaryotic cell jump from being a simple prokaryote to the large complex cell it is today? Well, endosymbiosis explains this cell fusion event. In Lawful Heart's 2013 compilation on endosymbiosis, the theory is described as a cyanobacterium being engulfed by a protist. Let's imagine a large cell looking for a meal to eat. The large cell finds a prokaryote to eat. However, the prokaryote, instead of being digested, escapes its doom. The prokaryote has now found a stable home while simultaneously providing benefits such as nutrients for the larger cell. In a Nature journal review of this molecular mechanism and physiological functions of endocytosis, endocytosis is described as an essential to neurotransmission and regulation of many cell activities. Therefore, endosymbiosis explains how higher eukaryotic life developed. Finally, we will talk about one particular important function of cells. The mechanism that cells use to bring particles into the cell is called endocytosis. Endocytosis is the active transport of particles into the cell. So here's our cell and particle. The cell begins to form a vacuole around the particle as it approaches. Once the vacuole fluid forms, it merges with the lysosome, which is when the particle begins to be digested. As you can see, this is broken down into 5 main steps, making it easier to remember. A study by the Medical Research Laboratory of Molecular Biology in Cambridge highlighted how endocytosis is fundamental to both neurotransmission, signal transduction, and the regulation of many plasma membrane activities, leading it to being essential for higher eukaryotic life. This study focused on clarithin-mediated endocytosis and how additions or substitutions in endocytosis allow for adaptability. So, to wrap up, the main difference to remember about prokaryotes and eukaryotes is that prokaryotes have membrane-bound organelles. Endosymbiosis is the theory that eukaryotes evolved from prokaryotes, and cells use endocytosis to bring particles into the cell using active transport. We hope you enjoyed our video. Our sources are below.